Hi, this is Mahalakshmi Sampagavi, Assistant Professor, Department of Pharmacology, KR College of Pharmacy, Bangalore. Today I wanted to explain a topic called as monoclonal antibodies. Antibodies are produced by a specialized group of cells called B lymphocytes. When a foreign antigen enters the body due to immune response, B lymphocytes develops into plasma cells and liberates antibodies or immunoglobulins of various types such as IgE, IgA, IgD, IgE, IgG, IgM. So now, what is an antibody? An antibody is nothing but a protein used by the immune system to identify and neutralize foreign objects like bacteria and viruses. Each antigen recognizes a specific antigen unique to its target. Monoclonal antibodies. Today's topic is nothing but monoclonal antibodies. Monoclonal antibodies are the antibodies that are identical because they were produced by one type of immune cells, all clones of a single parent cell, where mono means single. Coming to the polyclonal antibodies. Polyclonal antibodies are the antibodies that are derived from different cell lines. Then isotopes. Isotypes according to differences in their heavy chain constant domains, immunoglobulins are grouped into 5 classes or called as isotopes, which are IgG, IgA, IgM, IgD and IgE. Then coming in IgG, again it is classified into IgG1, IgG2, IgG3 and IgG4, blood and tissue liquid. Coming to the IgA immunoglobulin, it is divided into A1 and A2, where this is present in stomach and intestine. IgM antibody is nothing, it's normally pentamer, occasionally it is an hexamer, multiple immunoglobulins linked with disulfide bonds. IgD, where 1% of the proteins in the plasma membranes of B lymphocytes where the function is unknown. IgE on the surface of plasma membrane of the mast cells play a role in the immediate hypersensitive and defensive for parasite. Coming to the structure of antibodies, this you can see the diagram of the antibody, that is the structure of the antibody is shown as follows, where there is a history behind the monoclonal antibody, how actually it has been developed. So we should know a little history behind what do you mean by monoclonal antibody. In the year 1890, a scientist called as von Behring and Kitasato discovered the serum of vaccinated persons contained certain substances termed antibodies. In the year 1900, Ehrlich proposed a side chain theory. In the year 1955, a scientist called as Jerne postulated natural selection theory. Frank McLaughlin expanded. Almost the same time, Porter isolated fragment of antigen binding called as FAB and fragment crystalline FC from rabbit Y globulin. In the year 1964, Littlefield developed a way to isolate hybrid cells from two parent cell lines using the hypoxanthine aminopterin thymidine selection media. In the year 1975, a scientist called as Koller and Milstein provided the most outstanding proof of the clonal selection theory by the fusion of normal and the malignant cells. In the year 1900, Milstein produced the first monoclonal antibody. This is a brief introduction about the monoclonal antibody. Role of antibody in the immune system. How it is being showing its important role in the immune system. Each antigen has a specific antigen determinants located on it. The antibodies have complementary determining regions called as CDR. It is nothing but complementary determining regions. These are mainly responsible for the antibody specificity. Each antigen has several different epitopes on it. They are recognized by many different antibodies. All these antibodies thus produced act on the same antigen, hence these are designated as polyclonal antibodies. In general, naturally produced antibodies are non-specific and heterogeneous in nature. Hence, there are several limitations in the use of the polyclonal antibodies for therapeutic and diagnostic purposes. Thus, there is a need for producing monoclonal antibodies antibodies for different antigen. Coming to the advantages of the monoclonal, they are homogeneous in nature. They are specific to a particular antigen with a particular epitopes. The example is rituximab. It is used in the cancer treatment. Now coming to the cancer immunotherapy. Here immunotherapy is nothing but it's a treatment that uses the certain parts of the person's immune system to fight diseases such as cancer. This can be done in a couple of ways, means in the many ways. Stimulating your own immune system to work harder or smarter to attack cancer cells. Giving your immune system components such as man-made immune system proteins. The types of the cancer immunotherapy are monoclonal antibodies. These are man-made versions. Antibodies can be very useful in treating cancers because they can be designed to attack a very specific part of the cancer cells. Immune checkpoint inhibitors. These drugs basically take the brakes of the immune system, which helps it recognize and attack the cancer cells. Coming to the cancer vaccines. Vaccines are the substances put into the body to start an immune response against a certain diseases. We usually think of them as being given to healthy people to help prevent the infection. But some vaccines can help to prevent or to treat the cancer. These treatments boost the immune system in a general way, but this can still help the immune system attack the cancer cells. These antibodies are used to treat the cancer. The immune system attacks to foreign substances in the body by making large number of antibodies. An antibody is a protein that sticks to a specific protein called an antigen. Antibodies circulate throughout the body until they find and attach to the antigen. Once attached, they can recruit other parts of the immune system to destroy the cells containing the antigen. The side effects of the monoclonal antibodies are fever, chills, weakness, headache, nausea, vomiting, diarrhea, low blood pressure, rashes, etc. This is about the monoclonal antibodies. Few of the antibodies which are approved by FDA are as follows, which are used to treat in many type of the cancer, such as trastuzumab to treat the breast cancer, bevacizumab to treat the lung cancer, cetuximab to treat the colorectal carcinoma, panitumumab to treat the colorectal carcinoma. Thank you one and all.